forming front.
Unless you want me to not throw the card.
Sitting way up front.
Yeah.
Like you're interested?
Well,
Can you sit on the Can you sit in one so I don't clobber somebody else with my knitting?
good, I won't get clobbered.
Yes, I know.
Oh, yeah.
My daughter is a school teacher, and she's got another teacher who has a baby.
Great.
So I'm
I'm
helping her out.
Yeah, right, that's what it was. Yeah, that's right.
Oh, that's not fair. That's like digging the centers out of the chocolate.
Well, was that, were you with the PCs? Oh, right, yeah, that's right. So, because we had somebody else help us move stoves the other guys, and that's why I thought, you don't look like one of these guys.
Only because I haven't checked my email.
Oh, yeah, that's right. Now I remember, yeah. I know so many people lately, kind of swarms and everything, yeah. It is a busy time of year, for sure. This is like, we kind of barkeep and attendance and meeting people and everything kind of keeps the things the beats.
How have you been?
It's kind of funny because that's when everybody's like, oh, my people are crazy.
And then I went.
Well, so did you guys have
And it was your name.
Yeah. So,
and you're right in our neighborhood.
Yeah, we're on.
Yeah. Yeah, that's
and then just our topics and speakers. If you can hear it off. So that's where the network is, tries to
well, good. I'm glad you guys are into this talk.
typically
Usually I tell people, oh, yeah, what night was that? And then they show up like, yeah, right on. Yeah, good for you, Dan. That's cool. Well, that's a sign of dedication. We gotta do. Well, you'll really like tonight. We got two.
so that when it does open up.
Actually, I think he gets here soon. I gotta write something down. Oh, nice to meet you. Oh. No, I don't think so. Hey, Pat. Good. How you doing? Good.
Pardon. Thank you. I'm sorry? They wanted 100 chairs. I don't know where I can put them all. I'm kind of running out of room to put chairs.
Is that kind of what you were hoping for? Maybe not the rack. There's options.
At least they're on wheels.
Hi. I saw you caught a storm today.
Yes.
Nice. Where was
Hours
that at?
ago. That was in Small.
No way. So, and then it looked like, what were you filming?
Denver spots that I think were out in the field. And then let over and was
I'm like, what are these vibes there?
January was, you know.
Oh, it has just a white spot on it around.
Yeah.
Wasn't good.
Do you mind if I take
Am I still coming up tomorrow?
Yes.
One-ish?
Yeah. They didn't give me a specific time when it
Actually, I don't have any yet, but I am a garden educator.
Tell you what, I'm really interested in
Getting some hives at the school. Good, I'm beginning to work at.
what's wrong?
Saving and Wrigley right now. Yeah.
The one hole. We have two now.
Yeah, I'm sort of, I'm an urban farmer educator, but I don't know much about bees yet. And so I want to get kids.
But looked at it yesterday, and it's not quite built out enough here.
Where we can place them. It's really, really slow.
Yeah.
Or
Coaching house, the one
packaging.
Well, Lisa Weaver was one of our.
Unless we don't get aware of the frames.
Look, we have the person like any of them.
Then this week, really?
But what we have,
We were built out on quite a bit. The one I got today, 4 feet up on the table. Came down from the tree. The one tree.
Was this a swarm?
Yeah.
Oh. Tell me, what happened? How'd you know it was a good swarm? And how'd you know that it was?
It wasn't a big one. I mean, the cluster was, you know,
Yeah, like, and any full complaint you see, which is hard to monitor. But have visitors, about 20. And then the last, 4 or 5 days, there was nothing. And then today, it's like, holy smokes. Yeah. They're not going in the pot, they're going in behind the board. We've got some they're going to be. It's not here. When they're swarming like that, I don't think they're
No.
drop it into high.
No, all I have is a box.
Yeah.
And I just lift the branch and,
Well, then, but how do you, what
oh, then I brought it home and I don't have a word.
Oh, okay. So you, the whole hive, part of the hive, or leave 5 flames out, or what do you do?
Well, it's a word, so just the top part.
Oh, okay.
So I just take a couple out of them.
Actually, my grandfather. That's why you have those people that don't stay. That doesn't mean I'm going to try it.
You might have to try it. I don't.
No. Right?
Yeah, I don't know.
I could just sit.
Yeah, they're all on.
Hi everybody. I'm going to make just a quick announcement, and then we're going to get started here in a minute. But before we do, Andy Miller is going to be walking around with a survey that he's got. It's a paper survey, so if you have a pen or pencil, Andy will come through. Anybody who hasn't gotten one yet, would you just raise your hand and he'll give you one, a copy of it, and please complete it at the end of the at the end of the evening. If you could turn it back in or see to it that Andy gets a copy of that, or gets the copy back. There's a bunch of people on this side over here. Well, we've got a great meeting lined up tonight. We've got Dr. Ramesh Sagili, who is downstairs trying to find the front door right now. I might have to run down and get him. Or if there's somebody else that can go reading down there. If not, we also have Dr. Dewey Karen is back from Bolivia. We're really excited about that. So he'll be doing our Q&A in the second half of the meeting tonight. What else? A couple other things. I might as well go over this stuff right now. We've got some great raffle items tonight, so make sure you stick around for that. We have a library. The Portland Urban Beekeepers has a library. It's almost always going to be on this back table in the back of the room, on the right-hand side of the room. The library is, sponsored and paid for by Portland Urban Beekeepers, and you're welcome to check things out. And there's a simple checkout system, but it's lots of bee books, bee culture, American Bee Journal, and then some other bee books that you might want to use and check out and learn about the bees. So, our librarian is James Moore, who is, I don't see him right now, but, he's right over here. So, it's at the opposite side of the room. It's not where James is, it's on the other side of the room. So, help yourself to that. Let me see, what else? The raffle tonight, the raffle item is, we've got 3 pairs, I believe, Glenn, 3 pairs of tickets?
Yes.
3 pairs of tickets to the Portland Eco Film Festival.
No.
No.
I mean, yeah, but just the one film.
Just the one film, right. And it is the May 9th?
July 11th.
July 11th, I'm sorry. Which is, hang on, I'm looking here. July 11th is More Than Honey. Really good film. In fact, Glenn and Mike and I went to see that. I think it's done very well, and it's got some great information in it, so you'll be happy to win a pair of tickets to that if you're one of tonight's raffle winners. So make sure you stick around. The raffle will commence in the second half of tonight's meeting. One other thing I wanted to remind folks is that one of the members and myself, we're going to be putting together some beautiful professional quality postcards of bees and maybe some information. Mark Wenzel and myself, and these postcard sales, the sales of this, the proceeds, I don't know what we're going to sell them for. We haven't really thought through this too thoroughly, but the idea is the proceeds, 100% of the proceeds of these postcards will go to the Oregon State University Bee Lab. Ramesh, did you hear that? We're selling some postcards, and 100% of the proceeds of this will go to your lab.
Yeah, thank you very much.
Yeah, absolutely. So we're excited about that. So it's a way to contribute and actually receive something for your contribution. But we need that Bee Lab to be there, and we need Dr. Sagili to be working on research instead of writing grants. So maybe this will help him out a little bit. Okay, I got to check. Ramesh, I'm sorry. Is your stuff pretty close to ready to go? I got a couple more small announcements.
Yeah, I'm still looking. I found somewhere without putting any change in that. So
oh, you did? Okay. Can somebody?
I'll just plug your meter. Where are you from?
It's like towards the north side. It's a white.
You couldn't park underneath?
No, it was full. So.
It was full?
I'll just go down to him and plug his meter.
Okay.
You want me to come with you?
No. Is it a whiteboard?
Whiteboard. There's a bee sticker on the back.
That's what Dr. Sagili was trying to get in there.
Dr. Sagili, can I get your keys and I'll
Oh, yeah, sure.
I'll get it.
Oh, that's nice. Thank you.
That's it?
This-
And what am I looking for?
That was Gary Warren that just took charge there. He's the guy who is the executive director for this facility, so I think he can probably find a parking spot for it. So, just as a reminder, next month we will have Galen Beatty from the Columbia Land Trust and the Backyard Habitat Program. Is that right, Sydney? I think that. Columbia Land Trust and Backyard Habitat. With the Audubon Society, correct. She will be here with a wonderful presentation, and that'll be at the June meeting. So we'll looking forward to that. That's going to be wonderful. So anyway, without further ado, I'm going to go ahead and Dr. Sagili is in charge or head of the Bee Lab at OSU, and he is doing some research down there. Tonight's presentation will primarily be about some of the research that they're doing and also answering some questions with scientific backing, which is always good, so that it's not just anecdotal stuff that people think is accurate. That'll be good. Any questions will come in the second half of the meeting because you're certainly welcome to answer or ask questions to Dr. Sagili, but in the second half, as soon as Dewey gets here, I don't see him yet, but Dr. Curran is also back in town from Bolivia, and I know he's going to talk about some survey that I think he believes that he wants us to participate in, and also some of the work that he's been doing down there, but mainly lead the Q&A for us tonight after the break. So I think we're pretty close here. Mike, could you maybe bring down the light that's kind of shining on the screen?
Do you want me to give a real quick update with the finger stuff?
Okay, sure.
If you're ready, go with it. I'll do it later.
You just sensed that I was dancing up here a little bit, right? That's pretty good. So, yeah, there you go. Is that all right? Can everybody still see their notes? Because there's going to be a lot of note taking, I think, tonight. Yeah. I can also. Do you have a clicker? I can run it for you.
Oh. I have something.
Okay.
I can just touch pretty high really quick.
Okay, real quick.
Hi, my name is Sydney Mead, and I have a project that hopefully you guys have heard of. Last year we kind of rebooted Tour de Hives, which is a backyard tour of apiaries around the city of Portland. This year we're doing it again. It's going to be on National Bee Day, which is the third Saturday in August. So I think that's August 17th? Either the 17th or 18th this year. Yeah, so there's a couple ways that you guys can get involved if you're interested. Really looking for volunteers to help with the project. Always fun to have a team. We will need volunteers the day of. We also really soon will have information on our website about signing up if you're interested in being part of the tour. It's a great opportunity to also invite your neighbors if they're curious about what you're doing. A little goodwill goes a long way. We also are looking for sponsors. So those are kind of the ways you can get involved. More soon.
Excellent. Thanks, Sydney. It's really fun. Being as one of the people on the tour and also being a participant in it last year, I had a blast. And it's a great way to learn more and see other people's hives, which is always fun. Okay, let's do a little bit of, first, a couple of quick questions. Did everybody get a survey that Andy was handing out? It's an informal survey that we're doing here. I haven't even seen the questions, so hopefully it's not about, what your income is or anything. But if you haven't, then you can see Andy here. He's right here in the gray shirt on the center aisle, his hand raised. The other thing, I think one of the things that we could do right now is just, we have lots of new members, many of which are first-year beekeepers. And I think it's wonderful. And one of the things that we're hoping to do is to keep you as beekeepers and make sure that your experience stays positive and you hold on to your bees. So, Melissa, do you want to do a quick Zenger update? I can say that I had the most fun that I've had in a long time out at Zenger last week. We were walking through the apiary and looking for a swarm, and hi Lois. And Cynthia says, well, there's a swarm right there. And so we got to catch it and hive a swarm right in the middle of it while we were doing some other little task around there. And everybody had a great time. But Melissa can give the other details about the Zenger community.
Well, I think the biggest thing, since there's so many new faces in the room, is to let you know that there is a farm on the outskirts of town. It's in southeast Portland on 122nd and Foster. And it's called Zenger Farms. And out there we have 5 populated hives, but we'll have up to 9 or 10. And if you don't have your own way of getting the hands-on beekeeping, then that's why we're housing it's one of the reasons why we have the bees out there. So there is a committee that is mostly made of pub members. The core committee is doing the administrative planning side, and then we put out the email to the rest of the community to let you know whenever we have a work party. And unfortunately, because it's weather dependent and need dependent, we can't give you a calendar for the whole summer. So it's kind of with a few days notice. Usually we send a blast out to the listserv. So if you want to be a part of that, I'll be taking names back there. And there's also the Facebook. So the pub Facebook will generate, we'll put the post out there too. Any questions? Oh, and there is a kid component that we're starting to tease out in this.
I noticed on the event it looked like there was a once-a-month bigger meeting, but an administrative meeting, and it's not the meeting out on the farm.
Correct. Exactly. So the core committee, and if you're interested in...
That's good, because I might have driven out there and been by myself.
Yes. Okay. Thank you. If you have any questions, please don't hesitate to come and find me during the break. All right.
Okay, great. Thanks a lot, Melissa. Okay, Dr. Segilia, I believe, is ready now, and so we'll go ahead and hand over the mic. Do you want a lapel mic, or you?
Yeah, I tend to walk a lot, so.
This one, or? Okay.
you hear me in the back? How about my nice English accent? How many of you have heard me before speak? Oh, there are several. So. Okay, so it's another opportunity to have a nice English accent talk, right? So what I'll try to do, Tim, can we switch the lights off here on this side, or maybe dim it? I think my slide is the wrong one here. I think, okay. Sorry about that. Actually, the title I was having a different one, but I think I forgot to save when I saved the title slide. So what I'm going to do is talk a little bit about how many of you are very new to bees here. I mean, you just started bees this year. Just this year, okay. Not so many. I'll see that I'm not going over your head on information here. So I'll try to balance what I can here. So what I'll do is go over a little bit of applied biology. When we talk about honeybee biology, we start with basics from CAST and then go on different things. But I'll give you some applied biology, and then Tim asked me basically to talk about the research program that we have at OSU. So I'll focus at least half of the talk on that. But then I thought there might be some interesting things you guys may want to know as well. So I put some applied biology and research together. So what I'll do with this applied bee biology is go over a little bit of genetics. I know there is a lot of interest in queen. How many of you here are really into raising queens? Like you really want to? Okay, there are at least 4 or 5 of you. And yeah, that's a good thing, and that's an encouraging thing I have seen for the last 5-6 years. There is a lot of interest in raising their own queens rather than buying from Hawaii or Southern California or Texas or Georgia, right? And there are good advantages of it as well. We'll go over those as well. So what I'll do is very fast go over genetics, which is haplodiploid system, which is a very weird system that we have in honeybees. Have you heard of diploid drones before? How many of you have heard of diploid drones? There are at least 5 or 6 hands. I was expecting only 1 or 2. Now, I asked this question to the audience, and many of you, they said they have never heard of diploid drones. So, I mean, it's not just for fun, but I just wanted to tell you why diploid drones are important and what are the consequences if you have diploid drones. Then, how many of you have seen queens fighting? No one, right? You have? I mean, not in a video.
On the video.
Yeah. Have you seen 2 queens really fighting?
No. On the video.
Not on the video. Really, queens fighting. Yeah. No. Even I have not seen one. But I have a video which we made those queens fight. So some of you might have already watched if you have listened to my talk before. So we'll show you that video and what are the consequences and why queens fight and those kind of things. Then I'll go over a little bit about bee breeding programs. People are very interested in bee breeding and selection. I'll see. I don't think I have too many slides there, but I'll quickly go over that as well if possible. So honeybee genetics. So many of you know this is a haplodiploid system, right? So they have these males, the drones that you see here, these are the haploids. They have 1 set of chromosomes, right? But these are the ones which are more, we can relate to ourselves. These are diploid. Like we have 2 sets of chromosomes, like 50% of that comes from our mother and 50% from our father, right? So that's how we are diploid. So these guys are diploid as well. So they have 2 sets of chromosomes. The females I say here have 2 sets, 1 from our mother and 1 from our father. Whereas this drones, these are haploid. They have 1 set of chromosome and all are coming from the mother, right? So there is a joke about these guys. Some people say drones don't have fathers, they have grandfathers. Is that a true statement? Do you agree? Many of you, right? Those who haven't, I mean, those who are very new, I didn't want to confuse you with that statement, so I'll try to explain why I'm saying that. So the drones, these are haploid, right? And I don't know how many of you have heard of asexual reproduction or parthenogenesis. That's how they are produced. So the queen doesn't need to pass a sperm to produce these guys. These guys are haploid. They can be formed even without a queen mating. She can just release an egg and make these guys. And again, I don't have time to, I think Tim has given me only 50 minutes or an hour to speak. If I get into asexual reproduction, it may take another hour. So, but if you are curious and have questions, you can ask me after the talk. So these guys are formed in a very weird way, which is, I mean, not only honeybees, there are several other insects and some reptiles, some amphibians, they are also having the same system, the haploid system. So these guys are basically, that's why the reason why I say they don't have dads is, I don't know how many of you, I mean the new ones, the new beekeepers here, you guys know that the queen has to mate in the sky, right, with 15-20 drones. Probably your beekeeping club has already given you some update on the bee biology here. So when the queen mates in the sky with 15 to 20 drones, she stores the sperm for her lifetime almost. So she has a sac-like thing in her body, which we call a spermatheca. So all the sperm, millions of sperms, they will be stored in that spermatheca, and she can keep laying eggs for the next 3 or 4 or 5 years as well. So that sperm is required when you have to have these diploid individuals. So she has a very beautiful mechanism where she squeezes her spermatheca to release a sperm and the egg at the same time. So there is a fertilization process that goes on. So the egg is fertilized with the sperm, and then you get an embryo, and then that's how you get your diploid individuals, right? That's what happens in any animal that you can think of as diploid. You have to have a sperm and then an egg, right? Whereas in this case, so if she doesn't squeeze her spermatheca and release a sperm, then she lays only an egg without the sperm being touching that egg. So then these guys are formed, which are your drones or males. So when she releases an egg without a sperm, so where is the dad or the father? There is no father, right? Because there is no sperm involved in that. So there is no father. But still this guy has to have some male figure to relate to, right? Otherwise he's in trouble. So he has this male figure in grandfather, right? Because this queen had a father and a mother. So that father is the grandfather for this guy, and that's how he's related to grandfather, not her father, right? Not right. So did I confuse you, or are you still with me? Perfect. Okay. Yeah, if it's still confusing, sometimes it's too much information for those who are new. So this guy doesn't have a dad; he has a granddad. So that's how he relates to that. So coming from there, I'll try to confuse you a little bit more with the next slide here. So now I said these guys are haploid, right? The drones are haploid. Now I am throwing a term here called diploid drones. So I said drones are haploid. Now I am saying they are diploid. So isn't it confusing?
Yes.
So let me explain you why. So don't pay attention to this side. Just pay attention to this side. So when do you get a diploid individual? So diploid, just remember this part. Diploid means you have to have a sperm and an egg, right? So then only you get a diploid. If there is only an egg, you are a haploid. That means you are a drone. So here, just for example, so here we have sex alleles. There are about, at least people think at this point, there are 19 different variations of sex alleles. So when I say allele, that's another form of gene. Like people get their colors for eyes, blue, red, blue, brown or black, whatever color you can think of. So those are coming from some alleles as well, forms of genes. So in the same way here, with our honeybees, there is a sex allele, or the sex gene, just to make it simple. So there is dependent on those alleles. So if you have a queen here, so she might have several variations, but here, to make your life simple, here I have probably, like, this is a thing I stole from this website here, so that the picture is pretty good. So this is a queen here, this is your drone. So the queen, for just to simplicity, I have put only 2 here. So 2 of these alleles are here. And this is your drone. So when the drone mates with this queen, so there are different possibilities of you getting different things, right? If she doesn't release a sperm, then what do you get? drones. So you're getting all these two different drones. So they can have either a red or whatever color you see in the back, it's green or yellow, whatever you see, is another color you get here, right? So this is pretty simple here. So the complexity starts here. So now the queen lays an egg with the sperm. So I said they become diploid, right? So these are your workers. Workers are diploid. She can, they feed a different food to make a queen. So there is another complexity to the other level here, but I'm not going into that at this time. So for most of the workers that you see in your hive are produced like this, right? Your queen is there. You have a drone. The sperm comes from this guy. So it's stored here, and she releases the sperm along with the egg, and you get these workers, and they can have. So they will have one of this gene, the blue one here, and one from the red one from here. So you get a one type of worker here. So there will be another worker with probably this green and a blue, right? So these are diploid. So this part is okay here. So now I want you to focus on this side. So again, a same example, similar kind of these are different colors, because I said there are different variations in sex alleles. So suppose this queen has these two different alleles here, a red and a blue, and the drone is a blue guy. So what do you see here? These are not related, right? So this guy might be related to her. So he is a brother, or so here is an inbreeding thing going on. So this is a female, a virgin female queen that flies into the sky to mate with drones, and she finds some brothers as well they are flying. There is a possibility always. That's why queens mate with 15, 20 drones, so that they can reduce the chances of mating with their own. So here, the example what I'm showing you here is, so this guy is related to her. So this guy is a brother. So he is also flying in the sky along with other drones from different colonies, and he happens to mate. It doesn't happen quite often, but there is a good chance. So that's what I'm giving you some tips here, those who are interested in queen rearing or just for information. You need to understand why this is important. So this guy mates with this queen, and then, so the possibilities again, this is an easy possibility, right? You understand. So she did release a sperm, so you get a drone either with a red or with a blue or whatever, and these guys are okay. So this is the most important part here. So your queen mates with this guy and releases her sperm, the sperm of this guy, and then you have a possibility where you get a red and a blue. So this guy is okay. But here, if he shares, if the individual that is being raised shares the same genes from the drone and this blue from the queen, so this guy is in trouble here. So he is a diploid drone. So he is still a drone because there are two possibilities of becoming a drone. So if a queen doesn't release a sperm, you get a drone. And the other possibility is sharing the same genes, same alleles, then you can be a male as well. So that's how this guy became a male, but he is still diploid because there was a sperm that was shared. So he's in trouble. He doesn't understand now. I'm just joking that it's a diploid drone because and these diploid drones you will not see in the hives because the bees are so good at detecting these guys. They will eat them up or destroy these as soon as they hatch. So you will not see these diploid drones in the hives. But some researchers long back, maybe in the 50s, they have tried to raise them in the lab and they have seen how they behave. So these are not very useful. They may not be able to mate as the regular drone, and they will have several problems. So that's the reason diploid drones are eaten up. And the whole reason I'm giving you this story is just not for fun. There is some applied aspects to that. So have you seen something like this before? Those who are experienced, right? So and we call it spotty brood pattern, right? So there are several reasons. I'm not saying that this is the only reason why you see these in between for those who are very new to bees. So if you see a brood pattern like this, if you look at another frame, there might be tons of eggs. She has laid very well in each of those cells. But then you look at some other frames in the hive, and you find something like this. There are too many spaces empty like this. So if you ask a commercial beekeeper or an experienced hobby beekeeper, they would say, it's a bad queen, it's a spotty brood pattern, you should either requeen or do something else, right? So one reason for this spotty brood pattern is those diploid drones. If your queen was mated with her own, that is her brother, then you have these eggs that are being eaten by all these different workers that are realizing that these are not the useful food. So that's how you see all these things, the empty spaces in between. So that's one reason why a spotty brood pattern appears as well. Is that clear, or did I confuse you too much with that? Can we go to the next one? So that's the part about I know, honeybee biology is so fascinating. We can talk for 365 days and still there will be something left over. That's I always say that. I teach a bee biology at Oregon State, and it's a 10-week term. And the kids are not happy. They still want to learn, and it's done in 10 weeks. So that's unfortunate, but that's fine. And then I wanted to go over the selection and breeding process. Many of you know probably, but those who are new, you know that honeybees are not native to North America, right? We brought them to the United States in 1600s. So I'm going to go into this thing here, the bottlenecks. What are the problems that we have with the genetics? Because we have a very narrow genetic diversity in the United States with our honeybees. And that's a problem. You'll see that I explain in the next couple of slides here. So what are the bottlenecks? When I say bottlenecks, many of you have heard of this term before, right? Bottlenecks. Those who have a science background probably. If you are into engineering or something, probably. I'm not sure if you have heard of that. So these are bottlenecks are usually the limitations. So introduction to the new word. So we bought our honeybee from Europe, right? And there are species and such. Is there something I can write on, Tim? No.
A light.
No, to write something on like a board or a flip chart or something. No. That's fine. I can draw something in the air. So what happens is you have Apis mellifera, right? That's your European honeybee, or Apis mellifera ligustica, which we call Italian honeybee. Apis mellifera carnica. So Apis is your genus, then a species, and there is a subspecies as well, right at the end. So we have about 6 to 7 subspecies in the United States. But there are 26 different subspecies in the world for this Apis mellifera. So we have only like 1/4 of that genetics that we bought in from 1600s to whatever, 1922, when we passed a law called Honeybee Act. So we didn't want to import any more honeybees from outside because we were threatened with different diseases and pests like tracheal mites, varroa mites. Eventually they came into the United States, but it took 50, 60 years. So we could avoid 50 or 60 years getting those things, but eventually they are here now. So we are stuck with the genetics that we brought into the United States since 1600s up till 1922. So that's one problem. And then the introduction of mites. So the varroa mite, that probably the new beekeepers have not yet dealt with those mites. So those varroa mites, when they came into the United States, they destroyed most of the wild hives, the ones in the feral or in the wild. So those are the hives which were maintaining some kind of genetic diversity as well. So we lost all those as well because of this varroa mites since 1987 until 2000, whenever we got a little better with all these chemical treatments and several things happened in between. So that's what happened was the introduction of mites, they wiped out our genetic diversity as well because all the wild hives were dead. And then, this is the recent problem which we have is concentrated queen production. How many of you buy queens from outside? Outside means from a queen breeder, or most of you, right? Or you raise your own queens? Or you buy package bees, right? So package bees come with a queen, that's what I'm asking you.
Oh, they catch swarms.
Oh, you catch swarms. Perfect. That's a good thing. So most of the queens in the United States, at least I'm happy that you guys are trying to catch swarms and raising your own queens. But commercial beekeepers don't do that, right? They can't catch swarms all the time. So their bread and butter is all their bees. So they buy most of the queens from outside. So when they buy these queens, so the problem with genetic diversity here is they buy these queens from only a few different sources. There are not too many. Most of your queen producers are in the south. From California is major. Then you have some in Hawaii. You have Georgia, Florida, Texas as well. So there was a neat study from Washington State University that said that there were about 463 mother queens that were used by these all different producers to make 1.6 million queens that we use in the United States. Just imagine how much of a problem that is. So 463 mother queens, you make 1.6 million queens for the industry in the United States. So no one knows how that plays, and I'm not making it like sound it crazy that, oh, it's very serious, but I'm just telling you that that's a problem as well. So these are different bottlenecks that we have. Introduction of our bees from a different place because honeybees are not native to North America, and we have introduction of mites that killed most of the feral hives in the 90s when they were introduced. So that's a problem with, that's why I say why we have a problem with our genetic diversity is because of these three things. Then queen issues. Recently we see these queen issues as well. The quality. Some people are complaining that they have this premature supersedure. Most of you know supersedure. I'm assuming that no, for the new beekeepers, okay. So you know swarming, right? So when a colony gets to a bigger size, they swarm. So there are old queens usually going with 50% or 60% of your bees. So that's one thing. So that's one form. That's a natural form of reproduction for them, right? Swarming is a natural process. It's a good thing. Don't think swarming is bad. How can you catch bees, right, if they're not swarming for you? And it's good for them because that's a reproductive process for them. So that's how they reproduce. So that's a colony fission. That's what we use a term for that. So in this case, supersedure, if your queen is not in good shape, if she has a problem, maybe you can even try. You can remove a leg for your queen, and you will see they will supersede her. They'll make a new queen. So if your queen is sick for some reason, or you smushed the queen accidentally in your hive, so then they will make another queen. So usually that's a supersedure procedure. Usually it's related to health. There is an emergency queen rearing as well. When I gave you the wrong example of smushing the queen would be an accidental kill. So then they will make some emergency queens. That's a different thing. But supersedure is still the old queen is there, but she's not performing well. She's not laying well for some reason, and they make a new queen. That's called supersedure. Then there is this problem of some beekeepers are complaining that there is this early drone laying. So you buy a queen and it's beautifully laying well for next 6 months, and then suddenly you see she's not laying well. You see, have you seen that, Glenn? Mostly in some cases, maybe you're not buying queens from outside. so that's the reason Glenn is not saying those. So many commercial beekeepers also report that there is this drone laying pattern. So what happens? So she's running out of sperm. So she was not well mated. After 6 months, so what's the point? For first 6 months she was laying well. I'm just giving you an hypothetical example here. It could be 2 months as well. So you see the queen is beautifully laying and you're very impressed, and within 3 months or 4 months, then you see that it's only laying drones. You see the drone cells are bigger and they're protruded out, so you can see those differences for those who are new beekeepers. So this early drone laying is another problem. So these are all related to the quality of mating of the queen. So if the queens are not well mated, then you have a problem with that. So failure in requeening as well. Some people figured out that the queen acceptance is pretty poor as well. So the queens that you're introducing, the new ones, sometimes are not has anyone had that problem before? Like you put a queen and she has not been accepted yet. So there might be several things about the queen quality. So I'm just vaguely giving you some quality characteristics here, but there are several things. Even the drone pheromone release, if she's not well mated, then the physiology of the queen also matters. Then she's not releasing the pheromone in the required quantities. They have to release some queen equivalents every day. So they are not getting that as well. So these are some quality issues. So that's why with all these things, the whole point I want to make is local queen concept. I know here Tim, Glenn, and several people are really interested in having a queen rearing program. That's very important because it's not only that you have a locally adapted queen, but probably if you're doing it at the right time with your skills, maybe you're avoiding many of these problems as well. Okay, I need to come now, I guess. This is still I didn't have the edited version, but this is kind of a very crude version of the video fight that we have here. So here are two queens. As I said, you will not see queens fighting in a hive because they fight, but you are not there when they are doing that, right? So here we put two queens together because one of the queen producers, he gave us like 10 queens, and he said, we have extras and no one is buying now because it's almost September. So I said, okay, we'll do some research as well, and before we kill them for looking at nosema and other things, we thought let them fight as well so that we can have a video, and probably some of you will be interested in looking at it. So one is an Italian queen, this one, and don't there is no take-home message from here, but don't think that whoever wins is the best queen and you wanted to buy those or raise those queens. I have no preference for one over the other, but I'm just showing you how intense these fights are. So this naturally happens in a hive. It's not a thing that uncommon, but it happens in a hive. So for those who are very new to beekeeping, this is not like every day it happens like this in your hives. Whenever you have those swarming process when happens, then they'll make 10, 15 queen cells, right? So there might be 2 or 3 of them coming at the same time out of their cells, and they will try to find each other and kill because there is only one queen that is supposed to be in the hive. So this is called a queen dueling process, and that's what I'm showing you here. But for the most experienced people here, the reason for you it's not a new thing, but I thought there is an interesting thing that I wanted to show you here is, have you heard of spraying behavior before, Glenn or Tim? Spraying behavior, yeah. The queen sprays something onto the other queen. Have you heard of that before?
I've heard of that, yes.
Yeah. Yeah, let me show you that and then I'll explain when the spraying thing happens.
Is it a foul smell?
I didn't smell it, so I don't know whether it was a foul smell or not. But it's pretty attractive to the workers. So the workers will try to congregate on the queen that is sprayed on. And people think that's one mechanism to, I don't know, Dr. Karen is here? Yeah. Dr. Karen, have you heard anything more about it? I know the research was only limited. There is only one publication by Dave Tarpey that I could find. But
That's one, yeah.
Yeah.
Why does it take so long? Are they stinging each other or unsuccessful?
Next time I'll put an edited video for a minute. That would be sharp, right? No, so yeah, they're stinging each other and they sting multiple times. I thought maybe this was like a 10-minute thing. I'm not showing you the entire thing here, but it lasted about 10 minutes. So what they're trying to do is they're trying to sting each other multiple times, and probably whoever gets the most of the stings, he dies eventually. So
they can survive the stings?
Pardon?
They can survive a certain number of stings?
Yeah, and then that's a good question. Sometimes probably both queens die, and that may happen as well. Then you are hopelessly queenless in your hive. But usually that doesn't happen. So you will see only probably one queen dying.
So the smart thing is to be the third queen who's sneaking out, flying around the drums, right?
Well, yeah,
That's the winner.
that might be happening as well. Who knows? There are so many weird things happening in there. Oh yeah, sure. Let me. Did you guys see that she sprayed something out from here? So that's a spraying behavior. I mean, the reason why I'm showing here is not for fun, but again, there are not too many videos. I don't think you can find any video on the internet with this spraying behavior. I mean, I wasn't expecting as well. I asked my student to just record that, and it was happening in front of us. And so this spraying behavior is basically what this queen is trying to do is, I mean, it didn't hit this queen, but the intention usually what people are thinking is, so this spray, if it gets on this queen, then it's pretty attractive to the workers. They will come and just ball the queen, the other queen. And maybe that's a mechanism to kill the other queen. Maybe that's telling them that this is the winner and she's the one who is the losing queen probably. So we don't know those. I'm just speculating here. But that's a nice behavior which is not so common. So that's why I wanted to show you. That's called spraying behavior.
So they would go on the sprayed queen and basically suffocate her? Is that how she would die?
Yeah. That's the problem. I mean, she might die because of stings as well, but that's another additional way to suffocate the queen.
And the queen can stay and not die, but the female in the hive can't.
And I think this is not related, but this I thought, I get some kind of questions all the time, like, can we use the frames? If you have a data, how many of you lost some hives this past? Oh, okay, significant number of you. So yeah, I think Dr. Karen might be passing on those surveys. Are you passing those surveys today, Dr. Karen?
Yes.
Yeah. So he'll be speaking after me about that. So I don't want to take his turn to explain that here. So when you have these frames, some of you might be very curious, right? I get these questions. Is it something weird I'm asking, or do you go through these questions? You ask, like, can I use the frames again? Because all this colony collapse disorder and so many murky things are going on around that people don't know. And they usually ask, can we use those dead colony frames again? And again, I'm giving you an ambiguous answer, yes and no here. But you have to see when you can use them and when you can't. So I'm saying no if American foul brood disease, I know. For the new beekeepers, it might be too much information on what is American foul brood disease, but this is another nasty disease that you have to deal with. Not so often, but you have to be very careful about that because those spores are very resistant. They can survive 50, 60, 70 years as well in the hives. So the only way you can get rid of that is to burn your hive. You don't want to contaminate other hives with this. But you have very experienced beekeepers in your group here, so you can show them and probably they will be able to identify those things. And if not, you can send us a sample and we'll see. I don't do any culturing, but we can see if we can help. Otherwise, it has to go to a Beltsville lab in Maryland. They do all the testing stuff there. So yeah, in this case, you don't use any of the combs and not even the hives and even hive tools. I would discard most of the stuff that you have used. And this is the question that I get maybe nosema. Many people will have this problem, right? Nosema disease. And with nosema, I don't want you to discard your frames. I know they are expensive. You don't want to throw your frames, which you have bought for your operation. So there are some ways for nosema. Again, it's a murky thing here. Nosema, we still are trying hard to understand the two different species. Most of you know there are two different species of nosema, right? We are dealing with nosema apis and nosema serrana. These are two different diseases. For those who are new, it's a gut parasite. It lives in the guts of the bees and causes diarrhea or dysentery. So if you have that problem, then there are some things that you can do because two different species are different in their susceptibility to either freezing or to the heat or to the sun. So this would be the best thing. If you have frames that you think are suspecting that you had a disease with nosema and your hive collapsed, then you can aerate these frame fumes. You can put it in the sun, and some studies have shown that the UV light has the ability to kill most of the nosema spores. So, and again, it's not a 100% cure thing, but still you can try those things. And exposed frames, I mean, I don't know if this is possible for many of you, to expose your frames to 120 degrees Fahrenheit for a couple of days. So if you have such a facility in your garage or if there's somewhere you want to make that thing happen, you can try that as well. But then I think the one I'm missing here is freezing. You can freeze those frames as well. If you do all these two different things, probably you will be better off and you can still use your frames again. I'll not go much on this one because I think this morning when I spoke to Tim, he said there is a lot of discussion going on among this group about the small cell foundation that many people are trying to see if that works for reducing your mite loads. So I thought maybe I'll go a little bit on varroa mite life cycle so that it helps you understand what I'm going to talk in the next slides. So this mite, for those who are new to beekeeping, these are the ones which you have to deal with, right? For sure. This is a huge problem. Colony collapse disorder is not the problem. Varroa mite is the problem for you. So here is your bee with the mite on it, and then the mites, they sneak into this larva to lay eggs inside. Those mites can't reproduce outside. So for those who are new, these varroa mites, they have to stay on the bees for sucking their blood, and then that's how they transmit viruses. But for reproduction, they have to enter into a cell where there is a larva before being capped. So they find those larvae which are older stages, and then they find them, and then they enter into the cell. They sneak into this brood food, and then they stay there for a while until they cap it. So the bees don't know there is a mite sneaking in, so they cap it and they start reproducing inside, feeding on the larva. So I'm not going to go over all these stages, but that's how what happens. So the first egg she lays would be a male, and the next ones will be females, and the mating happens inside the cell, and eventually there will be the mother mite along with a daughter would come out of a normal worker cell, but if it's a drone cell, probably you'll see two more mites along with the mother coming out of the cell. So that's basically your biology. Can you go to the next slide? Yeah, here are some more pictures to just show you how they look like. These are easy to see for those who are new. This is a female. These are different stages of the same female. So we call them nymphal stages, deutonymph, protonymph, deutonymph, and then this is the adult here. So these are the male mites. Has anyone seen male mites before? You can see those. I mean, actually, it's not a big deal. If you have drone pupa next time and you think there are too many mites in your hive, open those drone pupa and then have a magnifying glass or just pay close attention. You'll find these guys. These are very small actually, but they are not crawling on your bees, so they die after mating. So usually you will not see them on the bees. So on the bees, the mites you see are the females. So these are males actually, and no, I mean, even I can't see them in the hive. So if I have to see, I have to open up those pupa that are infested. And if they are in the right stage where they have produced a male, then you will be able to see them. These are some pictures. Again, I don't think you need those. You'll see a lot of deforming virus. We call this parasitic mite syndrome. You'll see some larvae in trouble as well with some melting larvae. So if you see something like this, it's basically a parasitic mite syndrome. But again, this is not an IPM thing, so I'm not going to go into this. But if you guys want me to talk next time about most IPM, I can go. So these are some products that are available. I thought just to show a little bit of our research as well. I thought some of you might be interested. I'm not a big fan of asking you to use chemicals in your hives. I would be the one telling you to use minimal as possible. But sometimes probably you are not able to keep your hives alive without these things. So I just am giving you some things that are available. It's called Mitaway Quick Strips. It's a formic acid formulation. There is an ApiGuard. It's a thymol product. HopGuard. It's beta acids, 16% beta acids. It's from hop. So these are some formulations available. And there's a new insecticide, a new miticide that came recently. Has been used illegally in the United States for a long time, but now you have a legal product with a Section 3 approved. It's called Amitraz or APVAR, APIVAR, and it's a nice product as well. So I'm just giving you options here, but that's a new product. We haven't tested it yet here, but this is from the last year. So I'm just showing you a very small study that we looked at for the efficacy of these products. So this was the best one, which we found in our study was the Mitaway Quick Strips. Then ApiGuard, the HopGuard was the least, but HopGuard is a nice product as well, but the only thing is you have to use it multiple times. You can't just use once and say, all my mites are gone. You have to use it at least 2 or 3 times in a row every week to have a better control. And it kills mostly the phoretic mites. It doesn't kill the mites inside the cells, whereas this product, Mitaway Quick Strip, it kills the male mites inside the cells as well. But then it is toxic as well. This is not as toxic as this one. So there are both advantages and disadvantages of each product. So I'm not advocating one product or the other, but these are some results that we had. Then coming to the point that Tim was asking me to discuss a little bit about this small cell foundation. How many of you believe that small cell foundation really helps with controlling mites in your hive? Okay, one. Okay, good. No problem. Yeah. So no, there is logically it's possible, but I will give you. There are no studies at this point that have supported this notion. There is only one study I can think of. I think it's from Georgia, Keith Delaplane. He's a researcher and extension person in the University of Georgia, and they published a paper where they didn't find any. Actually, they find probably more mites than the controls, or the other 5.4 millimeter foundation. So for those who are new to this, you might be confused. What is this guy talking about? These different cell foundations. So the foundation that you bought this year, those who are very new to bees, so that's about 5.4 or 5.2 millimeters in width, right? If you measure with your ruler, probably you'll find that. But usually, if you leave your bees in trees or in a natural setup, they might make a smaller cell. They might make a cell which is 4.9 or 5 millimeters in width. So the whole point here is how this thing came into is, so the whole idea of using a small cell foundation is some people think that you will have less space. That's why I was showing you those varroa mite life cycle. It's because the mite hides behind the larva, right? So if you're raising your brood in those small cells, probably there is less space between the cell and the bee. So the mite is kind of suffocated or it gets killed. So that was one idea what people were thinking that small cell might help. The other theory was behind small cells was if you raise in a small cell your bee, if you are forcing your bees to be raised in those small cells, maybe they will have a developmental time probably a day or day and a half less. So how long it takes? 21 days, right, for your worker bee to come out if you have known your biology already. So the whole point is having a small cell will make your bee come out in 19 and a half or 20 days. So then if you are coming out early, that means the mite is not able to produce more babies inside. So that's how we think that probably mite loads will be reduced, but that hasn't been proven. I mean, again, that study has showed that small cell foundation doesn't help with mites at this point. That's the only study that I can quote. So probably some more studies have to go before even we can look at these things. But then I want you to think more about these things as well. So when you use a small cell foundation, that's one advantage. I gave you like in 2 different ways how you can reduce your mite loads. But isn't there a possibility that maybe we will increase additional cycle of brood, providing mites more opportunity to breed? Because you're reducing your every time you're reducing a day and a half, right? So look at for 6 months or 8 months in the long run, probably you're increasing one more brood cycle for those mites to breed on. So is that a good thing? I'm just throwing you some ideas to think. There is no answer at this point. Even I don't have an answer for that. And this is one thing. And the next thing I want you to think is also maybe putting selective pressure on mice will make them develop faster as well, right? It's an evolutionary arms race. That's what you heard about. So there are 2 things that are competing against each other. So if you're trying to make your bee a smaller one and come out sooner so that the mites can develop, so mites are not dumb as well. They will try to evolve according to that life cycle as well. And maybe even in the evolutionary arms race, you may create another problem with mites as well. So I'm just throwing some points to think about. It's too much to think, I guess. But for those who are fascinated with small cell foundation, I just wanted to. Is there anything else that you wanted me to talk about?
I don't think so. I think that answers it.
Okay. Can we go to the next one? And how much time do I have now?
5 minutes.
5 minutes?
7, 10.
I'll steal time from Dr. Karen maybe. What time is he supposed to speak?
We're going to take a short break, and then Dr. Karen will go on and follow.
Do you guys need a break?
No.
Okay. No, I'll try to finish faster now. Okay. I know I wasted a lot of time explaining things, but probably you don't need that much of explanation. So yeah, I'll take maybe 6 or 7 minutes, Tim.
Okay, that'd be great.
Yeah. So this is a national colony loss because he wanted me to talk about the losses, state of honeybee health as well. So I put this slide. Many of you might have already seen this slide. Actually, this was from Dr. Karen as well. So this is a number. Around 30% we have been losing, except for this 2012 was a better year with 22%. So 10 to 15% is a normal loss that is acceptable for commercial beekeepers. Beyond that is all a problem. So losing 30% is not fun. So they can be out of business in a couple of years if they lose at that pace. We'll go to the next slide. Yeah. So this is for those who are interested in the Pacific Northwest. Again, this is a survey that Dr. Karen and myself have been doing for the last 3 years. And so if you see, can you just click one more time on that? So that it will give you that one thing. Yeah. So these are the numbers. The blue ones that you see are the national numbers, and whatever brown or red you see is the Northwest commercial, semi-commercials, and these are the small scale, probably beekeepers like you. So your category has been losing higher number of hives than these commercial beekeepers, and there are several reasons. Again, I can't discuss those because of lack of time, but you can catch me after the break. So there was about small scale was 28% and 22% for semi-commercial and commercial in 2012. So there are several reasons. This is from the national survey. Yes, I have picked these numbers from. So if you ask these beekeepers in those surveys, what are the reasons why they lost hives? Again, this is not very scientific. They are just guessing on their the best estimate that they can give. So they're guessing and telling things. So you can see that queen failure is kind of on the top usually, and then mites are always there. Starvation can be a problem if your hive doesn't have enough honey to survive in the winter. That can also cause your colony to decline. So starvation is another one, and CCD was one of the probably fourth or fifth, probably in some cases. Colony collapse. Again, how many of you know about colony collapse disorder? Do you understand what colony collapse disorder is? Even I don't understand it. So colony collapse disorder is a mysterious thing. It's a disorder. The media has used this term so much that people think that itself is a disease. It's not a disease. It's a disorder. That's what it says here. Because bee researchers didn't understand what that phenomena is when someone said that all the bees are gone, like Dr. Karen says, nobody home. So that kind of phenomena, when that happens, and there are several symptoms associated with that. No adult bees, there is enough food stores, the queen is laying well, you see a lot of brood. So those are some symptoms that are associated with colony collapse disorder, but no one knows what exactly causes that. So at that time they coined this term, colony collapse disorder, because colonies were collapsing and no one knew what it is. They said it's a disorder. So let us find out what it is. And so that's how it started. So then you have all these, we can go to the next one. He wants me to finish fast. So I'm joking. So you heard about all these different funny things happening, right? People started talking about cell phone theories and and many of these have been discarded. So our bees at OSU, they enjoy cell phones here. So this is the take-home thing for the people who are interested in colony collapse disorder. So it's a multifactorial thing. There is no one thing that is killing our bees at this time. We think the bees' immune system is compromised because of these pesticides, fungicides that are being brought to the hive, and those are mostly lipophilic. That means they are attracted to lipids. So lipids means wax, right? So your wax comb can get all the pesticides in, and they can stay in your hive for 4, 5, 6, whatever years, and it can become a problem for your hives as well. So there is in-hive chemicals that beekeepers use, like the synthetics. That's why it's not good to use too many chemicals. They can stay in the hive and create a problem. There is a nutritional problem. We have all this monocropping going on. Beekeepers are putting their bees in just one crop for a month or two, and they're getting only one source of pollen, and pollen is their protein, right? It's important for them. So those are some problems which we have: migration, lack of genetic diversity explained. So all these different factors together are playing a perfect storm role, and they're compromising the immune system of the bee. And these diseases and pests are always there, right? Your mites and viruses. So they are the killers now. So they were not killing your bees earlier that easily, but with so many stress factors on your bees, now it has become easy for these guys to kill your bees. So they are becoming more virulent. So yeah, so maybe I have 4 more minutes, I guess. So okay. So there are these different areas that we are looking at honeybee research at OSU. So health, nutrition, and pollination. These are 3 different things that we are looking at. Go to the next one, Ken. Can we click one more time? Go one more time. Yeah, I put some funny slides here. Can we go to the next one too? One more. Yeah, so we are looking at all these different things. We are looking at pests and diseases. So if you are a beekeeper, if you are interested in sending a sample, you can. At this point, we are not charging, but if too many samples come, then I have to see if we can set up a mechanism for charging, because it's hard, because you have to hire people to do that, and it takes a long time. But until now, we have been doing it free for those who have been sending us samples. So pests and diseases, we look at varroa mites, nosema, tracheal mites, and then we dissect these glands in their heads to look at the nutritional levels. So it's called a hypopharyngeal gland, which is producing the food for the larva, for those who are new to bees. And then we also collect samples of bees, comb, and pollen and other things as well to look at pesticides and fungicides. So this gives us a big picture. Go ahead. So this is the kit that I bought a kit if you want to see, but so we'll see these vials in there. We have a questionnaire that goes with this. So you tell us whether you got a swarm or you got a queen or what your management practices are. So you send these things to us and we do the analysis. Can we switch to the next slide? These are some people that help us. I call them resting worker bees here, but they don't ever rest. So your honeybees don't rest, right? So this is just there might be several people missing here, but I have several brilliant people helping me in the lab to do most of the research and analysis part. And this is if you send me a sample, it will be posted here on this website. It's called honeybeelab.oregonstate.edu, and you can find, these are anonymous. If you don't want to be you want to be anonymous, then you can just use those numbers, and we just put those, and you'll find how many mites per. If someone sent me 303 bees, we say 3 mites in 303 bees, so you can get a percentage out of that. So that's a good way to look at varroa mites. So tracheal mites, we look at nosema spores per bee, and then we give you protein content in micrograms per bee as well. These are some results from, this is the overall state thing, but the ones I was showing you was basically as an individual beekeeper, you can glean into your information. But when I put together all the information here, so for 2009, this is varroa mite, I apologize, it's in red so you can't see that. So prevalence and intensity, these are 2 different things, right? Prevalence is just presence of that in how many samples you look at. If I look at 100 samples and if 85 of them are testing positive for varroa, then I say 85%. So, but intensity is more important. Prevalence, everyone knows, there is no rocket science here. Varroa mites are here and probably every hive has it. But it changes, you see, it varies between different years. And of course, it's not a perfect data either, because the samples might not be coming from the same people each year as well. So the intensities are important. 6.5% in towards the end of July or August is not a good number at all. 4.4 is also not so, but it's better than at least this year. So it varies as well. So nosema was kind of good information, because no one had looked for Nosema for 10, 15 years in Oregon. So when we started this study in 2009, we have been tracking the number of samples that are testing positive for Nosema. So in this year, at least, it was 48% that were positive. And then we get these spores in millions per bee, million spores per bee for the intensities. Tracheal mites, again, unless you dissect your bees, you can't find tracheal mites. They live in those breathing tubes. So we do those as well. And it's not a huge problem. Tracheal mite is considered a minor pest at this time, but it can be a problem if you have varroa mites, Nosema, and you have tracheal mites as well. Then it's a big problem as well. So then I have tried to correlate these things here. Again, correlation is not always a causation, and I just put some numbers out of curiosity here. So this is the survey that Dr. Kellen and myself have been doing. So we see this. I want to focus here, like see this colony loss percentage and look at the intensities. So it kind of correlates with that. So as it goes down, you see a percentage loss is also decreasing with this thing, right? Then it decreased in 2011. You have a pattern here. So I'm just showing you some patterns here. Can we go to the next one? Nosema, again, this is another disease that you have to deal with as a beekeeper. So there are two different species, as I mentioned before. Sometimes it's easy to find if you have a lot of defecation outside the hive in the front, not on the legs. In the front, if you see too many crawling bees and they're just defecating there is a good chance it's a Nosema. But you can send the samples, or if you have the ability to do at home, you can have a dissector, you can have a compound scope and try to put those samples in, and you can find those as well. So the spores look something like this. These are two different species here. Can we go to the next one?
Were you looking at the defecation there on the slide?
Mm-hmm. Yeah. So that's a defecation.
In magnification.
Oh, these are the spores. Yeah. So you can collect this and put it under a microscope in a water, and you can find this. Yeah. Or you can dissect your bees. So we take guts off. So if you send me bees, I take 100 guts off the bees, and then we grind them, and then it goes into a hemocytometer, and then we look at those spores in there.
Is Nosema something that they can cure themselves of?
Good question. Yeah, Nosema sometimes, it can be. Sometimes it depends on what kind of genetics you have in your bees, and sometimes without any problems they just get better. But in some cases, probably you may have to feed them fumagillin. So it all depends. Again, with the number of spores that I'm telling you here, it's very murky. I think no bee lab at this point has a handle on what is the exact sample size to tell you that, because not all bees are having Nosema. Remember that not all bees, if you have 40,000 bees in your hive, not all 40,000 will have the disease. Only there are a subset of bees that will have it. And then if I'm taking a sample and none of those bees that are infested comes, then I tell you that, oh, you're fine. If one bee, and there are again a problem is, there are some bees which are, we call them super infested. So one bee has 50 million spore per bee, and unfortunately if that comes into the sample, I'm giving you a wrong result as well. So the result that I give you, I try to be as forthright as possible. I'm giving you a presence or absence test. Rely on the data. If I say Nosema this many spores, don't worry about the number of spores. Just worry that you have a Nosema disease. So that's for sure we can tell. But I'm taking a new graduate student in the next couple of months, and he will be looking at most of these Nosema things which we don't understand. The prevalence, the intensities, those are very intense experiments he's going to do. And hopefully in next 2 years I might provide you some information on that. So this is the temporal phenology of Nosema in Oregon. If you're curious, this is not for hobby beekeepers. We're collecting samples from commercial beekeepers throughout the year. And if you see here, in May is the one month where you see a high spore levels. And this is here mean Nosema spores per bee in millions. So it just peaks up, and then, so that's what your question, coming back to your question, it gets better. So without any treatment, sometimes by June or August, you are not seeing many spores either in those hives. So it's kind of fascinating thing. We are still studying it, so I'll not take too much time to explain this. But so this is the last one with the bee nutrition. I'll quickly go over in a minute, and can we go to the next slide? So nutrition is very important. That's why I wanted to focus a little bit on nutrition here as well. This year was a bad year in many states like Texas, and many southern states had drought, even the Midwest. And here in the Pacific Northwest, we did relatively well, but still the nectar production was low. I mean, you talk to beekeepers, they didn't produce much honey last year. So that tells that nutrition was a problem. And whenever there is a nutritional problem, you will see bigger losses as well. And that's what we are going to see, more losses this year. I mean, this year means between October 2012 to April 2013. That's the time when most of these BIP surveys or what we do, we ask them to give the numbers between October and April 2013. So this is, some studies have already shown that nutrition is a big deal and you have to be very careful. It can be a problem for your colonilosis. Can we go to the next one? So carbohydrates are one thing that your bees need, that's nectar, right? But then pollen is the most critical one. That's what your babies that are the bees are raising need for their development. So that's important. Let me skip this slide. So protein can vary in pollens between 10 and 40%. And then you have to look for not only the total protein in your pollen, but also amino acids are very critical. There is a certain ratio of amino acids. Again, I don't have time, so I'm not going to go into that. But amino acids are very critical as well. Can we go to the next one? So yeah, pollen is important because you get, so beef, like you eat chicken for your protein needs or any meat, that's the only thing that bees eat, pollen. They can't get their protein from anywhere else. They have to eat that pollen. So if your hive doesn't have pollen, then that hive is in trouble in raising the brood. So there is a protein called vitellogenin, which is important for the longevity of the queen. So how long your queen survives or how long your bee survives is dependent on this protein called vitellogenin. It's a glycolipoprotein. That means it has glucose, it has lipids, it has protein as well. And this is directly correlated to pollen. If you don't have pollen, they will not have much of these stores of vitellogenin in their bodies, and your bee lifespan reduces based on that. Sterols. This is the most one I wanted to focus. Sterols are important. Like cholesterol. We have cholesterol, right? Is cholesterol good or bad? Both. Perfect. That was an answer I wanted. So too much cholesterol is bad, but you still need cholesterol. Otherwise, your cell development and all those things will never happen. So you need cholesterol in a certain amount. So bees need cholesterol as well, and it's called 24-methylene cholesterol that comes from pollen. So you can't just substitute that kind of cholesterol for bees. So when you feed your bees with those protein supplements that you get from bee stores, basically, I'm not sure whether they add some things like, does anyone make their own protein supplements here? No one? Okay. Have you seen anyone making, and you have a recipe for those? Oh, okay. So look for recipes next time. If there are commercial beekeepers that use that brewer's yeast, they put some canola oils, they put some egg yolk into that, and what else? Some sugar to make it attractive. So they mix those pollens. And I asked them, why do you mix canola or egg into that? And most of them have answered that they just found the recipe from another beekeeper and they're using it. They don't know why. So the whole point of those things they use in the hive, in those canola and all these oils, they have cholesterol in it. So they are trying to provide some cholesterol to the bee. But then those cholesterols bees can't use entirely because they need, like our liver makes cholesterol for us. But for honeybees, they have to eat from outside. They have to get, most of the insects, they get the cholesterol from outside. I mean, they have to eat something, a plant product like pollen, to get their cholesterol. They can't make their own cholesterol. So they are dependent. So that's why it's very important. So look for your hives when in August or September, when you're feeding that protein supplement from a store, try to buy the one which has some pollen in it. Some stores will sell you with 5% or 10% or 15% pollen mixed into that. So that's what I would suggest is have some pollen as well. If you have some natural pollen already in the hive, then you don't need to worry. But always it's not possible. Sometimes there might be a pollen dearth. You don't see much pollen in your hives. And without pollen, they can't raise babies. So pollen is very critical for them. A little bit. I'm not saying like they need probably 10 pounds of pollen. No. A small, maybe even 100 grams or 200 grams of pollen is enough if you have that protein supplement going on. Can we go to the next one? Yeah, I'll skip this. These are some fun pictures, but I don't have time. Let me skip so bees do all. So this is another study that we are looking at pollen quality on honeybee physiology and colony growth. So we are testing different types of pollen to see how the bees will be doing. We collect these almond pollen, cherry, meadow foam, wherever these commercial beekeepers go for pollinating. We want to see how these bees will do with these different sources of pollen. Can we go to the next one? So this is an ideal thing. I wish this is not from my hive. We again took a picture from this guy. But this doesn't happen all the time. You see how many different colors of pollen are here? So that's how, because bees don't know what kind of pollen they have to collect. They will collect whatever is available, thinking that, and they will collect different types so that there is a lot of diversity of that pollen in the hive. Because there might be one amino acid absent in this pollen, If they collect the other pollen, that amino acid might come from them. So they want to have a balanced diet. That's why they collect so many. Otherwise, they could just collect one type, whichever is convenient for them. So, yeah, can we go to the next one? So the monocultures are a problem because they are basically getting you a single source. I know you guys don't go for pollination, but I'm just giving you a scenario of a commercial beekeeper. So we are testing like these different pollens with a mixed source as well to compare a single source with 4 or 5 different pollens mixed together as well. So just some pictures here. I said almond, cherry, blueberry, meadowfoam. These are different pollens that we have been collecting. So we have a flight cage where we put these hives in, and they start feeding on the pollen that we provide them. They can't get anything from outside. So that's the whole goal, right? To provide the pollen that we have collected for feeding them. So we do several things. We look at the number of bees. I'm not counting each bee, but you have the squares where each square can fit 9 bees. So you get a translation to that number, and we do some protein analysis on those bees. And can we go to the next one, Tim? This we haven't done, but I thought this is a fun slide to show you. After this, I'm going to stop, Tim. We have 2 more slides. That's it. So we do this proboscis extension response to test the behaviors of bees, because prior nutritional status can change the behavior of the bee as well in some cases. Can we go to the next one? So we stick these bees in these holders like this and test them with different concentrations of sugar. So the bees, for those who are new to bees, their antennae are like your noses. You smell with your nose, bees smell with their antennae. So they can detect different concentrations of sugar. If you give them 10% versus a 30%, they are able to detect the differences between those. But pollen, they can't. If I give them a 10% pollen in a petri dish and a 50% pollen in a petri dish, they probably can't figure which one is good. They might go for both. But whereas for sugar, they can differentiate the differences between sugars. Can we go to the next one? I'll show you this cool video and then I'll stop. This poor guy is stuck in his holders now, and then we'll start testing them with different concentrations of sugar. Some people use this kind of test for testing their memory and learning as well. If your bee is exposed to a pesticides or something, and you might expect, let me allow you to watch this. So she's taking her proboscis or the mouth parts out. It's a very common assay. People do it. I mean, it's not, you can't glean much information from this, but it's one tool to get some information on different things, basically the behaviors and memory and learning tests you can do with these kind of things. So you can train a bee with, like you can train your dog with this bell. You can ring a bell and give the food whenever she comes or he comes. And then they get trained. Whenever you ring a bell, they will come running that the food is going to come. So you can do the same thing with bees as well. People have tried sending some kind of a smell onto their antennae. Like you can have a fan here. You send that smell on their antennae, and whenever the smell goes through their antenna, you give them a drop of sugar. So they associate that. So it's called associative learning. So they associate the smell with the sugar drop coming next. So you can leave them alone for 30 minutes, and then you blow the smell again, and they start sticking their tongues out. So if there is something wrong with their learning and memory, then they will not stick their tongues out. So that's one other way to test their learning and behavior as well. So there are several things. I don't think that bees are small. They have a milligram brain, but you can do some short-term memory testing for sure. There's a master beekeeper slide. I'll show that, and then I'll stop. The research part I'm going to wind up here, but I'll just show you some participants here. Master beekeeper. Oh, yeah. So I think most of you might have heard, and probably Glenn or Tim or someone else might have already mentioned about this. We have a Oregon Master Beekeeper program that we started 2 years ago. So I know some of you might be on a wait list as well, because we want mentors as well. I mean, we don't want the program to be diluted and so that you're not very satisfied. So we want it to be a very good program. And I know our volunteers are very much appreciated always, and we haven't found too many mentors around this area yet to accommodate all. So we'll try our best. So the whole goal for those who haven't heard of this program is it's an education and service program for educating basically beekeepers, and so that's what I say here, to improve and maintain the health of honeybees throughout Oregon through education and service. And I know you might have already most information, but if you want more information, email us. Carolyn, my technician, is kind of in charge of this program, and even Dr. Karen comes to your group quite often, so if you want more information, you can ask him as well. Okay, I'll stop here. Thank you very much.
Thank you. Okay, we're just going to do a really short break tonight because we'd like to get Dr. Karen on to do our Q&A. And thanks, everybody. And we might go a little bit long tonight. If you want to stay, just do it. If we go past a little 8:30, is that okay, Gary? Okay, great. So there's some good snacks. Again, compliments of Mark Wenzel in the back of the room.
Thanks, everybody.
This is empty. I don't have one.
Oh, perfect. How are bees doing? Good. Yeah. Yeah. Still strong. Yeah. Good. That's just how he was feeling. Mine are doing okay. I'm trying to catch a swarm in my box, but I haven't had any luck. So I'm going to have these next week with all the warm weather. But I'll try to steal a frame of eggs. Start saving time. That'll be my experiment to see how that goes.
Like last year, it just didn't happen.
I Okay, good talking to you. Hey Pat, how's it going? Oh my goodness. Right. They're doing well. But they cleared out they each had one group box to start with. So they brought them Sure. Oh, I read that thing. It's really out there too. You get so into it. It's a shamanic way of the bee. Right? I don't own it, but I
just finished that. I feel like
You said it's only 3 weeks, so you might have that problem in the future.
I might, but they say that if you build, the books I've read on top are, if they're building them straight, get them to do
sharp. They tend to stay that way. But they say they won't get nothing in their head.
So,
But, so I'm hopeful. And then so then I was like, well, since I'm in there, I might as well look for the things Bonnie told me to look. But I need to make a checkbook.
Yeah, I need to
Because
do
I was
that
so
too.
excited, and there were bees everywhere, and they were kind of mad. And because I didn't use smoke, because we didn't have any smoke with Bonnie last time. So I think next time either use sugar water or use smoke. As I got closer to the group now, they were pretty mad at me.
Yeah, well.
So, which was good. I don't know. It's good, I guess.
Well, yeah.
I mean, they were, you know, they were active. Because when we moved our feeder we built another follower board, with a little hole in the bottom, so the board with feeders can be through there, and so they can get into it. So when I open that take the lid off, I don't actually have to go into their part. I just go in the back, and they don't even notice me. I don't even use my bee suit now. They have zero interest in me whatsoever. They have zero interest whatsoever.
That's amazing.
But they're very happy. I think they're happy. So I went in, and I didn't know what I was doing. But I'm just so experienced. You know, I need to get over to New York High. I'm going to get down to the Bunny High to see, you know. So but now I know what I thought it was funny looking at the rain. I was like, I'm not taking but, and then I saw large, and I was like, okay, so that's good, so I closed it up. I never tried to find the
3 weeks into that, they take all that wax that has so much energy, and there's actually some brood in there.
Yeah, but still, well, no, I was wrong. I looked it up online. I was like, that's a funny looking one. It was all cat brood. It was all cat brood. It was like 3 branches of cat brood.
So they're building up that line of workers.
Yep, which I think is going to be their focus. Bunny told me not to worry so much if there wasn't a lot of cats coming. I mean, they've actually lost interest in their feeder. I only filled up like once a week now, when they were like emptying it almost the whole thing every day. That's a good thing too, because it means that they're finding their own source. They only will go to that if they have to. So they're pretty happy.
Yeah,
I think at least. Like I said, I didn't see the food, but I want Bunny to come up next time and help me get inside.
Yeah, she spoke of it. In fact, she's coming up Wednesday. We've got school for Junaria. Wait, and what? Thursday I'm going to go down and get my on Saturday. Yeah, I was there. Did you have it in your truck? No, because that would be a great show in Healthy Road. Yeah, it would be. Hi, guys, actually, I told her that I could bring it home today, but, you know, I'm so experienced. I really want I need help, you know, at the beginning. Anyway, she I'm going down Saturday to get it, Saturday night, and then she's coming Monday or Wednesday. One of the days. Yeah, so Tuesday I know we're going out to dinner. Oh, that's right. Yeah, with James. Oh, okay. We've been on this one.
Yes, yeah. Maybe he's not streaming. I don't know. Did you turn it off?
No, it's on.
I was going to say hi, podcast.
Are you ashamed of talking to Adrian about the logo design? Are you that James? Yeah. Okay. Well, today I'm going to play the practice.
Just email me or
what's your last name?
C-H-A-N-G.
If I get Andrew, I'll be like, what is that Andrew's number? Okay, so what is your email?
Here.
I think where I need help really is understanding between like the T-test and the Z-test. What is it? Why am I looking at this? Am I looking at some of the chart now?
Right.
You know what I mean?
Yeah, that's my cell.
I think we're being
Yeah, that's my cell and that's my email address. C-H-A-N-G and my last name and my first initial at ohsu.edu.
Okay, excellent.
Yeah.
Excellent, thank you. Nice. So it is
Everybody, if you can start getting your red on this right here, we're going to get back to the
It's a tough job. Okay, everybody, if everybody want to come back to their seats and eat their last cookie, return your survey to Andy, who's walking around. There's going to be another survey here in a minute that
More surveys.
Dr. Kern's going to deliver.
Andy Miller. Is this some of the same data?
Okay, so as I told you in the meeting invite, Dr. Kern has been in Bolivia for the last 6 months, I believe, and we're really excited to have him back as our lead Q&A guy and scientist. And without further ado, I'd like to hand the mic over to Dr. Kern.
Thank you. Appreciate that. We are going to do a Q&A. I do have a survey that Ramesh has talked about. We have been doing a survey, but in looking what Andy has, and many of you have filled out that survey, that has the basics of the information that we're after as well. And Andy has been very generous and said that he's going to share at least that portion of those first couple questions on the loss part. And so rather than giving you a separate one to do, appreciate your doing the one for Andy. So if you had overwintered colonies and you had some success, some were alive, none were alive, all were alive, whatever, if you'd have that information on the handout on the survey.
And I have to leave. I have to leave in a few minutes, so if you haven't had a chance to fill it out, just turn it in to Tim, and I'll grab it from him later. Thank you.
Frank's at the table there. I think he has some still too as well. So appreciate that very much. I will say that, as Ramesh indicated, we have asked the commercial and the semi-commercial beekeepers, and I have already somewhere over 100 of the small scale, the backyard or hobbyist beekeepers as well. So we are generating the numbers. You have heard some predictions about losses this year, our survivorship. The unfortunate information we have, that will be published this week. The Bee Informed, I hope some of you went on that. That is beeinformed.org. That survey closed yesterday. That's a national survey. We have well over 6,000 individuals that have participated in the national survey, and the results will be out this week. I'm doing a press release right now. And unfortunately, the numbers are going to be back up to those numbers over 30%, where we hoped that we were out of the woods and numbers had been down. As Ramesh showed, we've been coming down for 3 consistent years. Well, the numbers bounce back up again. So it'll be interesting to see the numbers that I have for the backyard beekeepers in Oregon very much mirrors what's happened in the past. In other words, the losses in backyards are indeed higher, almost double what is happening to our large holding beekeepers, the large beekeepers that have more than 50 colonies, and the commercials that have more than 500. So appreciate that very much. I did bring along a contribution for your efforts in terms of working with a calendar. Some of you do subscribe to Bee Culture. This is their Bees in the Hood calendar for the year. Some lovely photos and great information. And from across the pond, for those that are interested, the Bee Craft calendar. So I brought those along for the people that are working on that particular effort as well, and for the library. Gary is making a couple of scans. How many heft colonies? How many of you are in a practice of hefting colonies? Hefting means you use your other sense. That is, as you're looking at colonies, you lift them up. Hefting colonies. Find it useful, those that are doing it? What do you find most useful?
When they're light.
Food stores.
Food stores, right? Food stores. Others? When they're light, compared to, quote, normal. Right?
Yeah.
High hefting. It's a good practice to get in. And so what I have brought this evening is an article that is on the Be Informed website. This is beinformed.org. On the website is Honeybee Net. This is a national program of trying to gather that information and trying to make some sense out of it. That's using scale hives. If you've looked at scale hives appropriate for weighing beehives, you realize they're a bit pricey to do what you want to do. The other article that I'm putting on is from the Bee Craft. This is the English magazine, where the fellow is using a scale that you would use to heft a luggage bag. They only go to about 100 pounds, but we're lifting only half of the column. So you're lifting one half, and then you're lifting the other half. Most of those are in the $10 to $20 price range. So that might be an alternative. So both of those are being scanned, so that you'll be able to go onto the club website and download those articles. Honeybee Net is the national effort in the United States. OSU has participated in that for years. Then the other is bee hefting from Bee Craft. Second item that I brought, maybe to kick off some discussion, I hope you know about the website www.pollinator.org. This is beinformed.org. This is Pollinator, one word, pollinator.org. A number of you run into issues, we all do. In fact, I was talking with someone today about neighbors. Okay, this is one of the flyers that we prepared, one of our committees for the Pollinator Group, which meets annually. Inviting bees to your property, no fear of stings. So if you have one of those issues or the potential of one of those issues, here comes Gary with our articles. I appreciate it very much. Thank you. If you have one of those issues, you might think of that. You can download that from the website. Here is the Bee Culture. It's the February issue. Bee Culture is the national magazine. This is the one that has tattoo on the cover. That's actually someone's tattoo. Has a lovely article on tattoos if you are into that type of thing. But this is the article, Honeybee Net, by one of our people at the University of Georgia. And the other one that I indicated was Bee Craft. Gary's made a copy of that. Electronic hefting. I said beehive hefting is electronic hefting. Okay, that's using a rather inexpensive luggage scale. So there are some resources that you might find of use. Question and answers. Who's got a question? Maybe we find someone that could answer them. Anyone want to? Yes, question.
I'm just curious about the Honeybee Net. Is that related to the NASA project that they were tracking?
Honeybee Net is initially started as a NASA project, your tax dollars at work, a backyard beekeeper that was interested. I had a casual conversation with him about all these records that I had from the University of Maryland when I was there. And he then put the ABCs together of that and came up with this project, and it has really expanded since then. So yes, the short answer, yes, that is a NASA project. He no longer is working at NASA, so the big bucks have gone a bit along the way. This was related to trying to document climate change. Why isn't NASA? Why is our money in that aspect involved? It is with trying to document climate change. And he had the concept that we can use beehive weights, and when the nectar flow occurs in a normal year, which we can easily measure with a regular weighing program, that we could somehow use that as information on climate change. It's a gold mine. Bingo! He really hit it. And in our area of Maryland, for example, we have records going back to when I was in Maryland in the very early 70s. I was an inmate at the University of Maryland for 11 years in the 70s. And we had all those records. And since the 70s, now to the 2000s, the nectar flow in the suburban Maryland area has advanced by about 30 days.
Over a 40-year period?
Over a 45-year, roughly 45-year period. 30 days, a month. And we are able to show this with high scale weights. Incredible. There are couple neat studies this year. Someone went back to look at David Thoreau's records of flowering dates at Walden Pond in the Boston suburbs. Another one went to Wisconsin and looked at the records of flowering there and found that too is predictable. It has been advancing by a certain amount every year since, well, Walden Pond goes back to someone help me out. On Walden Pond by David Thoreau, 1840-ish? I'll take that. Yeah, something 40, 50? Very long answer, but yes.
I have a question.
Question.
So, I was going to ask Dr. Sajili this, but so the bees have been around for 30 to 40 million years. Maybe why are they not in North America? And then why were they not? And and how is the introduction of honeybees in North America? What kind of impact is it having on the native pollinators?
Okay, he's still here, Ramesh. Ramesh is going to want to duck this one. But a great question, an interesting question. Ramesh, why, after all the time, were honeybees not in the Americas? They were simply originated in Africa and it spread into Europe and in the Middle East.
You want to answer? Or?
No,
You're putting me on the spot? Okay.
I'm putting you on the spot.
Yeah, that's a good question. I mean, we have to study the geographic, maybe how the continents were formed. You have to go that back to figure out those things. I mean, bumblebees were there. It's not just honeybees were not around here, but we had native insects like other pollinators like bumblebees and other mason bees and those things that you see. They were native here, but yeah, as Dr. Karan said, they were probably still there is a debate, but we still think they have originated in Africa and then they have spread to different continents. But I mean, that's a very good question. Maybe it's a continental drift, or we don't know what exactly might be the reason why we didn't have honeybees here, but other ones, because they had to go from somewhere. The origin is Africa, so they spread to Europe and then like that. So, but it took human help to come to North America. They didn't fly that hard. Like probably they were close from Africa to get to the European continent, but America was so much apart from there. Maybe that's the reason. I don't know. Is that something, or you have a better answer than that?
Well, I have one other piece to add to that. There are currently, depending on which expert you're looking at, 11 species of apis on planet Earth. 10 of those are in Asia, roughly in Southeast Asia, extending all the way over into Tibet and India, one of the mountain species. 11 current ones. The one that we rely on is not native to Americas. It was brought here, we believe, by the early colonialists. That's Apis mellifera. There was, however, an apis species based on the fossil record in North America. It no longer is here. So there was social apis species, at least one species here, that no longer exists. That record goes back to, most of the bees we think came from wasps. They developed hairy bodies because their diet changed from meat to vegetarian, and they coincided with the explosion of the flowering plants. So if you recall your history in terms of when flowering plants, that is other than the pine trees and grasses, etc., when they developed about 100,000 years ago. And so too, all of our records in terms of bees. Now we have some bees that do go back further than that, but the record for the apis species in the Americas, it was actually from New Mexico, and it goes back to about 20,000 years ago. Why it went extinct in that time, from that time, we don't know. Now your second question is a toughie. With our bringing this species to the Americas, what has happened to the native flora? Has this bee displaced? Has it made other species go extinct? What's basically happened? We don't have a direct answer for that, but if we look at the population of bees that we know as Africanized honeybees, they were brought in the 1950s to Brazil, and and they have replaced Apis mellifera from Europe. So the only bee that I keep in Bolivia is the Africanized honeybee. The only bee I can keep. You just can't keep others adjacent to it. We've looked at that bee, Dave Rubik, particularly at the very fine Smithsonian facility in one of the islands of the Panama Canal. Initially, he published a couple of papers indicating that some of the existing social bees of the Americas, what we call the Melipona and the Dragona, the stingless social bees that were here long before honeybees were introduced. Initially, he found, in looking at flower foraging, that when Africanized honeybees moved into an area, the social bees disappeared on that particular flower that he was looking at. And so he actually ended up publishing a couple papers. Arrival of Apis mellifera, the Africanized honeybee, is displacing bees, native bees, social bees in the Americas. However, that turns out not to be all of the story. The real story is what has happened with the honeybees are pigs. They are the pollen pigs of the world. They just go to anything and everything. So what happened as they came into the Americas, they did in fact push the stingless bees off of some of their flowers that they were visiting on. Unfortunately, Dave Rubik picked on a composite, a weed species, for his first study. And on that composite, before arrival, stingless bees. Following arrival, no stingless bees, just the Africanized honeybee. So on the basis of that, he said, yes, they're making it go extinct. It turns out the stingless bees just are not competing directly with the pollen pig, with our Apis mellifera. They are still there, they're still doing okay, but not on that introduced weed. The genus name is Baltimore, if you want to look it up. It's a composite flower, yellow, nondescript, sort of real weedy pollen flower. So, a very long answer to say, we do not know. In addition to problems with honeybees right now, we're having trouble with bumblebees. What that is related to, our population of bumblebees are tanking in a number of places. But that's not because we brought in honeybees. That's, we think, a large part of what's going on with some of our wild bees, our native bees, is loss of habitat. And in fact, that's what happened with Africanized honeybees, loss of habitat for the stingless bee. So no, there is no one study to point to, no one issue to point out that because we brought another species, Apis mellifera, to the Americas, something else disappeared from the record. And our way to get at it is looking at the bringing of the population. We initially thought yes, but we now think no. So it's a very long answer saying, I don't know. Is that what you were after? It's an interesting, certainly an interesting question. We could look at it as another aspect. What about the leafcutter bees? Our mason bees. What about the alkali bee? The growers of alfalfa from Utah north into Canada rely on those populations of bees to pollinate their alfalfa. Not honeybees. Honeybees don't do a good job on alfalfa north. They do okay in Nevada and south, but not north. What about when you put out all these sheds and have thousands upon thousands upon thousands of leafcutter bees living in these sheds, going into the drilled bores, into the tunnels? What does that do to the native bees that certainly would have been there before? Or in the case of alkali bees, it's a special soil condition. That's why they're called alkali bees. You can build an alkali bee bed and actually make one of these things. What happens when you make one of these beds? And so now in the soil, just you got hundreds of thousands of these one species, this alkali bee, nesting in the soil, going to alfalfa. What's happening to the native bees that should have been there? That type of research is doable. In other words, we could go to an area and set up that type of experiment. It hasn't been done, but I suggest that perhaps there are other issues when we do something very strange like bring a species from another continent to this one, or a population, the Africanized bee to Brazil, for example, or augment the populations of the leafcutter bee is not a native bee. That's a bee from India. The alkali bee is a native bee, but what about when we build one of these beds, or 2 or 3 beds around an alfalfa field? And their numbers go from hundreds to hundreds of thousands. What in the world happens to the other native bees that were there as well? And why are we not doing that? Because, as we found with pear orchards, canola fields, almond orchards, there are not enough of the native bees. There are not enough of the native bees to do the job. That's why this is neat, by the way. Put in one more plug. Inviting bees to your property. That doesn't say honeybees. And is not related, is not just simply restricted to honeybees. So that's another neat one about other bees. Yes?
Could you talk about super sisters and half sisters and the impact on the health of hive?
Super sisters and half sisters in the health of hive. I could, but Ramesh is an expert on this particular aspect. Super sisters and half sisters.
So you guys don't want me to go to Corvallis back? I need to leave now. So are you asking differences between those? How the super sisters are made, or what's the question? Curious about.
Yeah, I'd just love to know your explanation of the distinction between them and
yeah, like
how you would identify them.
Oh, identification is hard. You have to do a genetic analysis for those. Yeah.
What are they?
So the first question, the basic question. See, you're at the different level than them right now. So super sisters are the ones. Have you heard of that? There is a relationship, like 75% relationship between. So what happens is, let me give you a basic genetics here. So I talked about those haplodiploid genetics and all that stuff in honeybees. Was you the question? No, you were asking the question. So what happens is when you have this this diploid haplodiploid system, and then you have this altruism. We think like why did bees? I mean, whenever an individual organism is there, it's good for them to have their own progeny, right? Why do they allow the queen to raise babies? Why don't the workers start laying by themselves? So they're not related. I mean, if you see, so that's called this guy Hamilton. I don't know if you have heard of that Hamilton hypothesis. You try to Google. I mean, I can't give you the entire answer right now, but I'll try to answer as best as I can. But you can read more about that. There is a guy called Hamilton, Hamilton, Hamilton hypothesis. So this guy tried to answer this, the kin selection and all these problems that we talk about. So why do honeybees like the queens are laying? Why not workers? Because workers should also compete with the queen, right? They should say, oh, I have to increase my genetics. Why would I want to allow the queen to lay an egg rather than me laying myself? So if you look at that, the best hypothesis that he came out with was, if the workers are laying, then you're probably 50% related, right? Like you, as a diploid organism, you are related to your parents, maybe to your mom 50%, your dad 50%. So whereas so the super sister thing will come soon. So when you have your queen laying your eggs, so what happens is if she is mated with so there is a 75% max, you can see 3/4 relationship. So if a queen is laying, and if suppose there are some genetics, so we talked about that same drone, if she is related to that drone that she's mating with, those kind of inbreeding things. So then you are getting 50% from the queen and 25% from the male, right? So there is a 75% relationships. So that's a max you can have in a colony or any possible individual. You can be related to that individual with 75% of the genes. Whereas if you are trying to do your own job, then you are doing only 50%. So that's the best thing they can have in a colony. So that's a maximum relationship that you can see. So those are the ones which we call super sisters, which are 75% related to each other. So that's going back to your question, but did that answer your question as well, or you? You want more explanation on that? So you can create this, actually what people do is I order sisters. Like sisters, basically if you have a mother queen, you're grafting, those who are into queen rearing. So if you take a mother and then you are grafting from that colony, then you are getting sisters, basically, right? Suppose you make 10 queens out of the same mother's individuals that she has raised. So you're getting sisters. But there is a way you can make a super sister as well. So you make those things with a drone that you know of. You take the sperm. So you have to do, they can do it. You can do an artificial insemination. And then you can get that super sister as well. So it's a little more complex to understand. But is there another question to it?
We believe that it's very important that the queen have a diversity of male partners. And it can be anywhere from a dozen to perhaps as many as 24. That diversity means when we measure things in colonies like a better winter survival when she has a diversity. It means better resistance to some things such as nosema. There are a number of qualities that seem to imply that multiple matings of a queen with multiple drones is a good quality. But when that occurs, that means within the colony, the offspring, the workers, may have the same father, or they all have the same mother, or they may have a different father, depending on the donor of the sperm. So at any one time in a colony, we get these subgroupings that have a higher degree of relationship to each other. It becomes critical. It's an interesting aspect, and it's very important when we get into the aspects of queen breeding, but it becomes critical when we're trying to do things that with our bee colonies that normally aren't done, like replace the queen with another queen, move brood from one colony to another, like move adults from one colony to another. And there, this relationship and who the father may be comes into play, because colonies that seem to have a large proportion of related sisters are generally not very accepting of a new queen coming in, where colonies that have a mixture of several dads with the same mom seem to be more susceptible coming in. How do we tell all of this? That's the hard part. You have to do it with gene sequencing. You have to develop markers and find out from that. So there's no easy way. But perhaps you've noticed when you look in your colony, some of the workers just look different than others. That's related. That's what you're showing from several fathers. Or the drones, look at the drones. Okay? Drones, one drone to another, just look very different. Okay? The entire inheritance from a drone is from mom. Drones have no inheritance from dad. From grandpa, yes, but not from dad. And so that particularly shows up in terms of the drones. And then that relates to, okay, in colonies where we're rearing queens, we want to increase the number of drones. Whose drones? Which drones? Okay? And so what we tell queen producers is you've got to have drone mothers, drone-producing colonies that just put out lots and lots of drones. And if you're doing that, you've got to have a mite control strategy because, as you heard Ramesh say, female mites prefer the drone brood. They can tell the difference. They prefer the drone brood, and they generate more babies, more females, by going into a drone brood compared to a worker brood. Okay? So all of these issues are sort of related.
Question over here.
Glenn, no, I'm not. I need an easy question. After all of these, we need an easy question. I'll come back to you. Bee sting. No. Bee sting. Not mine, not Glenn's. Anyone have bees that sting?
No, when I'm cleaning up frames, and there's a lot of pollen left, sometimes with mold on it, white mold on it, and so if I scrape that off, then I find bright yellow pollen. And then sometimes the pollen, or maybe it's nectar on top of pollen, that went through the winter, but the bees died out. It doesn't have mold on it. So the question is that pollen of any use?
Okay, the basic question is moldy frames. Moldy frames that are filled with pollen. Sometimes you see mold, sometimes you don't. If you have dead bees from a dead out colony, often you do see mold on that. Is that pollen of any use? The short answer is pollen is preserved as bee bread. It's not actually pollen. It's been converted by a lactic acid reduction. So it's not exactly the same thing. So it does have some livability, but bee temperatures being what they are, around 95 degrees, that high temperature, that high constant temperature tends to degrade the protein portions of that pollen. So what we usually think in terms of pollen stored over the winter, it has lost about half of what, at least initially, it had as nutrient value, perhaps more. Now, if it gets moldy on top of that, is that hastening the degradation of the protein portions? Short answer, we don't know. But what you're doing, that is brushing off or feeding that moldy comb to bit by bit to strong colonies. Don't throw a whole moldy box on top of a strong colony, but feeding a couple frames now, feeding a couple frames to another colony perhaps, let them clean up a couple frames and feeding some more is the best strategy we have. We don't know, most of these molds are not things that necessarily are pathogen molds for bees. So that's the good news. These are mostly plant pathogens, not honeybee tissue pathogens.
But they'll consume the bee bread, or?
Will they then consume the bee bread? If anyone has had any experience with it, they will do what they darn well want it pleased to do. Sometimes they will consume it very rapidly. Sometimes you'll look in 6 months later in the fall, and it's still there, and those same cells.
Well, do you have a recommendation then on what to do?
Feed it back. If you want to brush off, you feel better brushing off the moldy comb, mold, that's fine too. If you've got dead bees, you at least brush off the surface bees with a bee brush. Drawn comb is your most valuable piece of furniture in that colony. It is for the bees, it is for us. So any of those that we can rescue, preserve, refurbish, we can't, the bees can refurbish, we're ahead that much. There's nothing really directly to say that that's probably harmful. Yes, Matt?
Both of you had today talked about the survival rate of overwintering in commercial hives versus hobby hives. Why was there such a dramatic survival rate?
Survivorship, why in the backyarders is there less survivorship than seemingly in the commercial? For a couple of reasons. One of the ways is artifact of the way we collect that. Most of the larger beekeepers are in their colonies and doing things with them in January and February. And so they are finding colonies that are under some sort of a stress that if left on their own, if nothing done with them, would be probably dead in April. But they're rescuing them in February with their treatments. So that is part of the factor, certainly with that. The second is we are unwittingly being partners in killing our bee colonies. We don't mean to do so, but we do so. Moisture kills our bee colonies in Oregon, moisture. And commercial beekeepers have that aspect of moisture under much better control than we do in terms of our backyard colonies. They're proactive. They're finding those colonies and doing something with them prior to April. We're looking out the back window. Gee, they're alive that nice day in February. Gee, I saw them flying in March. Then you get your bee suit on and the smoker going finally in February or April, and you get out there and no one's home. They're all dead at that point. That colony could perhaps intervened earlier. So that's a large factor. They're doing something, not waiting. Yeah.
So how are they getting into their hives when it's January and February and it's 20 degrees out?
That's what you do as a large-scale beekeeper.
That's the risk you take.
That's the risk you take, yeah. The other issue is, as Ramesh and I are doing the surveys, virtually all individuals that have more than 300 colonies have their colonies in almonds in February. And of course, that's a few hours south of here. The weather is better. Almonds happens to be one of those very lovely pollen sources that just push colonies along. And so that's part of the issue. They don't have to get their colonies through to April. They only need to get them to February. They're in California. They may not all own their own truck. They're putting them on someone else's truck, or they're subcontracting with someone else. That's why you have a large number of these beekeepers selling nukes. They have to do something. They have to do something in April. They'd lose them all to swarms in the trees. They'd all be in the trees.
So is the key for us then feeding pollen over the winter?
Great question. Is the key for us feeding pollen over the winter? Yes and no. It's work. It's expense. Okay? And if you feed pollen, you are likely to build a stronger colony. Do you want a stronger colony in April? Do you want to chase the bees out of the trees? You want to go after swarms. So there is a risk. There's a risk in stimulating colonies early by feeding in February because bees actually don't eat pollen. They don't know what pollen is, and they don't certainly know that soybean is a very good protein source. They know nothing about it. So what do we put into that pollen that we feed them? Sugar. We trick them into eating this darn stuff. Okay? Incidentally, they're getting the protein, but they're really after the sugar, so we're really tricking them. So we're stimulating. We're pushing them along. So there is danger in stimulating colonies. That is strong colonies. A follow-up? You're like you.
So then what do I do?
They are your livestock, and how we want to then feed or not feed, stimulate, what we put into them, within some limits, are pretty much personal decisions.
And in the city, it's different. There's just pollen sources pretty much 12 months a year now. Is it enough? But the Oregon State Beekeepers Association used to have, I don't know that they still have, a contest to submit the first picture after the first of the year of bees bringing in pollen. And it's usually within a day or two of January 1st that somebody takes a picture of bees bringing in pollen.
Yeah.
So it's just different.
So when you say the danger is making them stronger because they will then swarm, you're increasing the chance.
Stimulating colonies will make them stronger, and so you may have a colony that gets a little too strong for the amount of space that they're in, and then that leads to swarming. Swarming is not a single causative factor. There are a number of factors. One is the amount of space, but also the population. So there are several interrelated factors. Stronger colonies, no, you will not necessarily make them swarm, but stronger colonies have a greater tendency that they are in fact going to swarm.
The first swarm that we got this year, Jim and I got, was March 30th out of a bee tree. So, obviously nobody was feeding that.
So I know virtually nothing about any of this, so my next question is, when they swarm, do all of them leave and then you have no bees at all? Or do a bunch of them leave and then some of them stay and then you still have the ones you have? Or?
Good question. In terms of swarming, swarming is colony reproduction. It's a division of the existing colony. So normally in a swarm, the primary swarm contains mom, the old mated queen, and a proportion of the workers. The proportion may be normally as much as 2/3 of the adult worker population. And they engorge on honey, and they take honey with them. Bye, Ramesh. Have a safe trip. They leave behind all the wax and all the babies and all the stored food that they don't take in their honey stomachs. So it's a colony division, colony reproduction. Colonies may also then swarm again that same season within another few days. Those secondary or after swarms no longer have mom, the old mated queen, but they are swarming with usually several of the new virgin queens, one or more virgin queens. So after swarms can have virgin queens. Then in the summer we get what we call swarms, And particularly also in the fall, we get what we call swarms. They look like swarms, they smell like swarms, but they're not. They are absconding, colonies that are leaving home. In some bees' minds, they're going to invade another colony. They're trying to try to usurp another colony. But in other cases, they are abandoning home, absconding. So that made a very simple question very complex, didn't it? But basically, swarming is division of an existing unit.
I think we need to kind of get ready to wrap it up. Dr. Karen's got some things that he needs to take care of, I believe. And I'd just like to take this moment to maybe no more questions for the night, but some of the questions we can ask on Facebook site. And those of you who are not a member of that, please join it, and because there's some great conversation going on, and a lot of the questions can be answered by 250 people or so. But the other thing is that Dr. Karen and Dr. Sagili are, we can push those questions onto them and maybe get answers to your questions and then feed them back to you.
And thanks to Gary, our host, our generous host every month. But thank him for making copies, and these are then be available. Honeybee Net and electronic testing. Thank you. Appreciate it. Good luck this month. This is a busy month. Thank you for doing that survey. If you haven't done one, you did have some overwintering and would like to participate, thank you. We will get that information back to you as promptly as we can. Thank you.
Thanks a lot, Dewey. We're going to do the raffle real quick. So if those of you want to participate in that, we're going to have three pairs of tickets to the Honey. Let me grab it. Honey, More Than Honey. Okay. And also, there is an announcement that we'd like to make here on March 25th. There is going to be a protest march, an international organizing chapter of international protest against Monsanto. And I know many of you are anti-Monsanto people, and those of you that are, you may want to know this is March 25th at 11 a.m. in this area. And Gary, who is the president of the Lloyd Center District here, has just wanted us to make aware of this. So this will be in the back of the room.
We'll post it on the Facebook site and on the events too.
I said May 25th, I meant May 25th if I didn't say May 25th. What did I say? March. Sorry, my bad. These are in the back of the room if you want to participate or even just go find out what that's all about. And then the raffle. Oh, okay. Okay. So I'm going to continue on here. That's, I think, about it. This is in the back of the room. Oh, they haven't been collected? Yeah.
You're doing the raffle
through the raffle through the phone. We've got a new electronic raffle thing. It automatically picks a name. Can you help me with that, Mike? James? It's electronics and it's high-tech, and so of all the officers in the group, I'm probably the least qualified for doing that. James? Oh.
Gotta help Grandpa program the
Yeah.
VCR.
James just said he's helping Grandpa program the VCR. I just like to say I resemble that remark.
It's a Betamax.
Yeah, Betamax. And I actually am a grandpa. 3 months and 2 days.
Anyone want to see these addresses? You're welcome to look at write the electronic addresses down here too.
Dewey, did you want to? Do you want me to post a survey to the site too? Your survey?
No, we want to do the survey this time of year. We don't want anything.
We want to end it. Okay. So we'll get all those.
Yes.
Okay. Okay. This is pretty cool. And the winner for the first pair of tickets is Jennifer Platts. Is Jennifer here? Woo, Jennifer! Jennifer, no Jennifer. So going once.
Yeah, we should say that this is a select program James made that picks from our membership list. It doesn't pick for people who are present. So Tim's gonna count to 5.
So it must be present.
Nobody answers.
Yes, that's right, it's on the right down to your name.
Yeah, must be present.
And these, your name will be on the guest list.
Yes. So, so far we have no winners.
So let me just refresh. Mm-hmm.
This is really cool. And it's refreshing.
Takes a second.
Takes a second. James wrote this program, so he's pretty proud of it. And the winner is Judy Morton. Judy.
Oh boy.
How many people are on this list and how many people are in the room?
Yeah. I'm going to just go grab somebody here in a second.
Yeah, I want to go.
Yeah. It's an awesome movie. Mike and Glenn and I saw it. I thought it was really. Yeah. The next winner is Sandra McQueen. No. This is really slick, but we might go back to the names out of the hat here in a minute.
Pick one out of the hat.
Casey Meredith? No, I know Casey's not here. Oh, God. This is pretty cool. I'm going to do this little dance that I do here. I just left. Janet Walsh. No, I know she's not here. I mean, I should just... I'll just...
Just pick one out of the hat.
All right. Never mind. This is so...
Oh well.
And the winner is...
Jake.
You pick it out.